Guys, what's up? Today we are coming with a very interesting topic which is called mitochondrial DNA. So DNA, the full form of DNA is deoxyribose nucleic acid. So before going into the DNA, the genetic material of this mitochondria, we must know that what is mitochondria. In eukaryotic cells, there are a number of cell organelles such as chloroplast and all this. So mitochondria is one of it. So mitochondria is a cell organelles. But according to the symbiotic theory, it is said that in the course of evolution, mitochondria was an entirely a different organism, a prokaryotic organism. So during the course of evolution, it start making a symbiotic relationship with the eukaryotic cells or eukaryotic organisms. So slowly with the time, it become a part of eukaryotic cells and become a cell organelles. So what this mitochondria do is, it is called the powerhouse of the cell because it generates ATPs. So with the process of oxidative phosphorylation through the electron transport chain, it makes ATP from oxygen and simple sugar, which is called glucose. And at the ultimate product of electron transport chain, or ultimate product of glucose glycolysis, Krebs cycle, and then ultimately it gives ATP, which is used for an energy source for all the function of the body, except the brain and use the RBCs. It do not use ATP, it directly use glucose for their energy purpose. So coming to the mitochondrial DNA. So generally the mitochondrial DNA is located in the matrix of the mitochondria, inside the mitochondria. In the short form, it's called mtDNA or mDNA. So the mitochondrial DNA is circular. It's like this, and it is double stranded. So it has two strands. It has only 16 kb in length, and it contains 16,600 base pair. It's haploid in nature, which is single, for it's not diploid. So it codes for 37 genes. From these 37 genes, 13 genes provide instruction for making enzyme involved in oxidative phosphorylation. I already discussed this oxidative phosphorylation, which is a process which uses oxygen and glucose to create ATP, and which is used for the energy purpose of the cell. So this is the structure of mitochondrial DNA. This is the structure of mitochondrial DNA. It has only 22 tRNA transfer RNA and 2 rRNA, which is 2 ribosomal rRNA coding genes. So it contains only exon. There is no introns in between the exon, like splicing in general DNA. There are some intron present in the mRNA, so it need to be spliced before protein synthesis. So in mitochondrial DNA, it is not present there. So in mitochondrial DNA, there is called a loop-like structure in the DNA. It's called it D-loop. The function of this D-loop is in the replication of this mitochondrial DNA or transcription of the mitochondrial DNA, mRNA, mitochondrial DNA. The starting point of the replication and transcription are the D loop. So it's called a displacement loop. It is little displaced as compared to the other strands of the DNA. So what's the difference between the nuclear DNA and the mitochondrial DNA? The nuclear DNA is the entirely genetic material which is present inside the nucleus of the cell. So mitochondrial DNA is an organelles DNA which is present in the matrix of mitochondria. So it is generally diploid, two sets of chromosomes. Diploid, the chromosome is like this. It has two sets, like 23 chromosomes, 23 chromosomes in human, it's 22 somatic and 1 sex chromosome. So it has two sets, which become 44 plus 2 XX or XY. So the mitochondrial DNA has several copies, but it is only haploid in nature. The inheritance of mitochondrial DNA is only from the mother. The inheritance of the nuclear DNA are from both mother and father, that is maternal and paternal inheritance. So it can discriminate between individuals of the same maternal lineage, but the mitochondrial DNA do not discriminate the individual of the same maternal lineage. So it is double helix, it's like this. The nuclear DNA is like this, it is helical structure, it is double helix, and it is not circular, it is linear form, and it is also double helical, but it is circular. This is free of envelope, but in nuclear DNA there is an envelope inside the nuclear envelope inside the nucleus. So DNA is packed into the chromosomes or chromatin, which we call chromosomes, and the DNA is not packed into any chromatin structure. This is the structure of mitochondrial DNA and nuclear DNA. So mitochondrial DNA has number of subunits. For example, this is the subunit for ATPs. These are the subunit for NADH dehydrogenase. This is the subunit for tRNA, cytochrome C oxidase, and 12S rRNA, 16S rRNA, and this is the nuclear DNA, which is linear and double helical. It has AA base pair, GC base pair. There are in both the DNA there are the the nitrogenous base pair, which is adenine, thymine, guanine, and cytosine. So coming to the topic, homoplasmy and heteroplasmy. Homo means same, hetero means different. So homoplasmy is a term which generally used to describe any eukaryotic cells whose copies of mitochondrial DNA are exactly same or replica or identicals. So heteroplasmy में क्या होता है, बिल्कुल difference होता है. Heteroplasmy is the presence of more than or number of mitochondrial DNA which have different shapes or different copies. So the presence of more than one type of mitochondrial DNA. So generally the number of copies are exactly same, but when there is difference, it may cause any disease. Maternal inheritance of mitochondrial DNA. Coming to the topic, maternal inheritance of mitochondrial DNA. So the maternal inheritance what is maternal inheritance? It refers to the transmission of gene from mother to the offsprings. So generally in during fertilization, the sperm contributes only 23 chromosomes. So okay, let's come again. The maternal inheritance referred to the transmission of genes only through the females from the mother to the offspring. So in higher animals, mitochondria shows only maternal inheritance. So the sperm only contribute 23 chromosomes. Mitochondria of the sperms are located in the neck of the sperm. So the head portion go only inside the nucleus of the ovum, so the head portion will get destroyed during fertilization, so the mitochondria will get also destroyed during that time. So only mitochondria is available of the mother, so it is of maternal origin. This is the structure during fertilization. You have seen this. This is the structure, for example, cell membrane, nucleus, and maternal mitochondria. So this is the structure of sperm. All the things are in the head portion, only mitochondria is in the neck portion. So during the time of fertilization, the sperm head get inside and the neck do not get inside the nucleus of the ovum. So the mitochondria are destroyed, and the only cell, the sperm contribute only the 23 chromosomes. So coming to the mutation level, so there is a question why the mitochondrial DNA has very high rate of mutation. So the mitochondrial genome has a very high mutation rate, 10 to 17 fold higher than the observed nuclear DNA. Why? It's because there is no repair system in the mitochondria. If there is only little repair system in the mitochondria, DNA repair system. So in nuclear DNA it always repair, but there is insufficient DNA repair system in the mitochondria. So there is lack of protein such as histone. The nuclear DNA are actually like this, because it is coiled on a histone protein, but the mitochondrial DNA lacks that. They do not have any histone protein in the structure. So the spontaneous error arising during DNA replication. So during the time of DNA replication, the spontaneous error will also occur. So that's why the mutation rate is higher in the mitochondrial DNA. So these are some disease which are associated with the mitochondrial DNA. So cyclic vomiting syndrome, Carinae-Serine syndrome, cytochrome C oxidase deficiency, like syndrome, mitochondria complex third deficiency. These are all the disease. These are diseases which are associated with the mitochondrial DNA. Thank you so much for watching the lecture. If you think that it is nice, or if you feel it is good, please like, share